Okay guys, hello and welcome. Assalamu alaikum to all of you. Okay, so in our lesson today we are going to discuss the Alliance High School trial paper, okay, specifically biology paper 1. It's going to be an interesting one as usual. So on to our first question. The diagram below represents an organism. Study it and answer the questions that follow. Okay, so let's look at it. So this is clearly a unicellular organism. We also have the contractile vacuole there. Okay, these are present in unicellular organisms that are found in aquatic habitats. Now this actually is specifically Euglena. Euglena is a member of kingdom Protoctesta. So we've actually answered part A. Identify the kingdom to which the organism belongs to. This is Protoctesta. Just a reminder, when writing the names of kingdoms or any other taxonomic unit, you are supposed to start with a capital letter. Part B, name the structure labeled X. Okay, so X is a flagellum. Now remember, it's pointing to 1. So if you were to write flagella, that will imply it's more than 1 and hence you will be penalized. So the answer is flagellum. Part C. Identify the type of nutrition carried out by the organism and give a reason. Okay. Now, when it comes to members of kingdom Protoctesta, these are broadly divided into 2, where you have the algae and the protozoa. Now, examples of algae, Euglena. You also have Chlamydomonas, Spirogyra. All of these have chloroplasts, okay, and because they have chloroplasts, they are capable of carrying out photosynthesis. So their mode of nutrition is autotrophism. Now, on the other hand, we have protozoa. Protozoa include the amebia, the paramecium, the trypanosome. These are not autotrophic, okay? They cannot manufacture their own food and therefore are heterotrophic. Now, in this case, since this is a Euglena, the type of nutrition will be autotrophism. Reason, presence of chloroplasts. Question number 2. State the phylum where all members have open circulatory system. Okay, this is Arthropoda. Now, another reminder, we are starting with capital A. And also, sometimes students mistake Arthropoda for Anthropoda. There's no N here, so it's Arthropoda. Part B. State advantages of the closed circulatory system over the open circulatory system. Now, in closed circulatory system, you are going to have blood flowing at a faster rate due to high pressure. So, such blood is capable of circulating over longer distance. Now, animals that have a closed circulatory system tend to be more active, okay? This is due to the efficient transport of gases and nutrients. Now, I would like to say this. I have seen books whereby they state one of the advantages of closed circulatory system is that it separates oxygenated and deoxygenated blood and therefore they do not mix. Now, this is somewhat true. For the more evolved systems in birds and mammals, yes, we don't have mixing of oxygenated and deoxygenated blood. But let's just pause here. Closed circulatory system is present in chordates, okay, in members of phylum Chordata, where we have Pisces, the fish, we have Amphibia, Reptilia, Aves, the birds, and lastly, Mammalia. Now, when we talk about some of these classes, like let me give an example of amphibians. Amphibians also have a closed circulatory system, but they have a 3-chambered heart, okay? Their heart has 3 chambers. On the upper, you have 2 auricles. On the lower, you have 1 ventricle. So a total of 3 chambers. Now, what happens in amphibians is that you are going to have a little bit of mixing of oxygenated and deoxygenated blood. Remember, you're going to have 2 auricles, but only 1 ventricle. So they do try to minimize this, okay, within the ventricle by having a shunt-like system. But a limited amount of blood, deoxygenated or oxygenated blood, tends to mix. So my point is this: when it comes to the advantage of closed circulatory system, as preventing mixing and of oxygenated and deoxygenated blood, it's true, but only applicable for some and not all the classes. Okay. Question number 3. Explain why malaria cannot be transmitted through blood transfusion. Now, when you talk about malaria is a vector disease, okay? So in this case, the vector for malaria is, of course, going to be the mosquito. Within the mosquito, you are going to have the Plasmodium parasite, okay? So this is the one that is responsible for transmission of malaria. Now, when it comes to the Plasmodium parasite, it requires the mosquito in order to complete a crucial part of its life cycle, okay? Without the mosquito, the parasite cannot successfully mature into the form that is injected into a new human host, okay, into a human. So essentially, it's a vector disease, okay? The parasite is transmitted from an infected individual to a healthy one through the bite of the female Anopheles mosquito. Now, I would like to say this, okay? Because this is not just revision, but we also want to build on our knowledge. This question has a statement that has been disapproved currently. Okay, what do we mean by this? Like, scientists are now aware that malaria can be transmitted through blood transfusion. I know, it's contradicting the statement, right? So if, for example, you get such a question, you're going to answer it the way I have stated it. But let's talk about currently, what are we aware through our research and so on, through the research of scientists. Now, the parasite that causes malaria, the one that I've talked about, Plasmodium, is present in the red blood cells of an infected person. Now, if an asymptomatic person, okay, a symptomatic person is a person, okay, that is infected with a disease. So, for example, in this case, they are infected with malaria, but they show no symptoms, okay, of illness. Wako sawa tu. Now, if such a person was to donate blood, the parasites can actually survive in the stored blood. Yeah, scary, right? And they can be passed directly to the recipient during blood transfusion. In fact, the parasites can survive for several days or even weeks in the stored blood, okay? Even when it's refrigerated. So this is especially serious. Actually, it's more serious than from a bite, because when a recipient, okay, when a person is infected this way, the parasites are injected directly into the bloodstream, okay? They bypass the liver stage. When you learned about malaria, there was a stage that involved the liver, right? Okay. When it's injected, when you have blood transfusion, the parasites pass directly into the bloodstream of of the recipient. So this can actually lead to a more severe, okay, onset of the disease. So that is just a by the way. Nowadays, yes, we are aware that transfusion of blood can actually infect malaria from one person to another. Okay. Let us proceed to question number 4. A boy held a locust upside down and attempted to drown it by immersing the head in the water. Okay, so the locust was held upside down and only the head was immersed in water. Explain the observation made. So the question is simply asking, will the locust die or not? And if not, what's the reason for this? Now, the locust did not drown, okay? The locust did not die. Now, reason being that when it comes to the locust, insects in general, their gaseous exchange system is the tracheal system, which consists of the spiracles, the trachea, and the tracheals. Now, spiracles are openings that are located on the thorax and the abdomen. Thorax and abdomen, not the head. When spiracles are open, they allow gases to pass in and out of the locust's body. So in this case, with the locust, having its head immersed in water, it's not going to suffocate because gaseous exchange is going to continue just normally, since spiracles are located on the abdomen and thorax, and therefore, it won't be affected. Okay, question number 5. Explain why malaria is resistant to quinine. Quinine is the drug that is used for treatment of malaria. Now, as stated, the parasite that is responsible for malaria is Plasmodium. So, Plasmodium undergoes mutations and develops resistance due to prolonged use of quinine. Proceeding to question number 6. The diagram below shows an experiment that was carried out to investigate a certain biological process. Study it and answer the questions that follow. Part A. What is the aim of the experiment? Okay. Now, if you look at the diagram, we can clearly see that a portion of the tree has been debarked. Okay. The bark has been removed. Now, with the removal of the bark, you are also going to remove the phloem tissue. So you are going to interfere with translocation. Translocation is simply the process whereby manufactured food substances are transported from the leaves, where they are manufactured through photosynthesis, to the rest of the plant. So the aim of the experiment is to investigate translocation of food substances. Name the tissue removed in the above experiment. That, of course, is going to be the phloem. State how the above named tissue is structurally adapted to its function. Okay, so we are talking about the adaptations of the phloem to translocation. Point number 1. It has companion cells that have numerous mitochondria to provide energy for active transport. It also has cytoplasmic filaments, okay, for which facilitate movement of food substances along. Part D. Draw a diagram to illustrate the observation made after 3 weeks. So that is our diagram. As you can clearly see, we are going to have, the upper part of the bark just immediately above the debarked part being solid. This is because of accumulation of food substances. The food substances cannot be translocated downwards due to the removal of the phloem tissue, and therefore will end up accumulating at that part. Question number 7, part A. Name the protrusions found on the posterior end of the skull that articulate to the atlas. Okay, so the atlas is the first bone of the vertebral column. Now it articulates, okay, on the anterior end with the occipital condyles. Okay, so at the base of the skull, we are going to have these protrusions, rounded surfaces that are known as occipital condyles. These are going to articulate with the atlas. So this leads to the formation of a joint that allows us to nod our head. Part B. Give the name of the joint formed in A above and state its function. Okay, so the type of joint that is found is a hinge joint because we can only move it at 180 degrees Celsius along one plane. The function, of course, is it facilitates nodding of the head. Our next question. Explain natural selection using principles considered by Charles Darwin. So Charles Darwin proposed the theory of natural selection that is based on the following principles. Number 1, variations exist between individuals of the same species. That means that within individuals of the same species, such as humans, you are going to have differences that are present. Okay, now what will happen is that due to competition of resources, you are going to have individuals of the same population struggling for existence. Organisms that have advantageous variations, okay, these are the variations that enable them to adapt better to their surrounding, are going to survive, grow to reproductive maturity, and pass on these characteristics to their offsprings. Those that have disadvantageous variations are, they will die and will be eliminated from the species. Okay. Question number 11. The diagram below shows the position of an image formed in a defective eye. Name the defect. This is myopia. Now, myopia is what is known as nearsightedness. This is a vision condition where a person can see objects that are close very clearly, okay? But objects that are further away usually appear blurry. Now, the main cause of myopia is that when the eyeball is too long, when the distance between the cornea and the retina is too great. So what happens is that the light is focused in front of the retina instead of directly on the retina. Remember, the retina has photoreceptors, okay? So light is supposed to be refracted exactly on the retina for the image to be perceived. In people who have myopia and the cause is that their eyeball is too long, light is focused in front of the retina. As you can clearly see, the light rays are converging, before reaching the retina. So it causes images that are distant to appear out of focus. Now, myopia can be easily corrected by using concave lens. These lenses are also referred to as diverging lens. So they cause the light to converge precisely onto the retina. Okay, so I have already answered part B. On to part C. Draw a sketch to demonstrate how the above condition is corrected. There we have it. So with the introduction of the concave lens, what we can clearly see is that the image is focused on the retina. Question number 12. 3 stems of tradescantia of equal length were placed in 3 solutions of different concentrations. The setups were left to stand for 30 minutes. The results were recorded in the table below. Okay, remember we have 3 solutions. Each of these solutions has its own concentration. So solution A, when the stem was placed in it, the stem had a length of 37 millimeters. After 30 minutes, there was no change. So it was still 30 millimeters. Now, we are talking about osmosis here, movement of water molecules. And that is the reason why we are taking the length of the stems before and after. Okay, we want to know if the stems have remained the same, if they have increased or decreased. Now, this is based on the movement of water. So with A, since we're not seeing any change in the length of the stem, that implies that solution A was isotonic to the sap vacuole of the tradescantia stems, and therefore there was net movement of water in and out, and finally no change in the length. Now when it comes to B, initial length was 37, final length was 36.7. So we have a decrease. The decrease in length is because the cells have lost some water. Cells will lose water if they are placed in a hypertonic solution. Remember, movement of water molecules is always from a solution that is hypotonic to one that is hypertonic. So if they lost water, that means that the cell sap was hypotonic to solution B. Okay, hence we have a decrease in length. Now, what about solution C? Initially, we had 37. Final length was 38, an increase. Okay, so this means that solution C was hypotonic. And that is the reason why water molecules moved from solution C to the sap vacuole, causing an increase in the final length. Okay. So part A, identify the nature of solution A. This is of course an isotonic solution. Part B, account for the observation made in solution B. So solution B was hypertonic to the cell sap of Tradescantia. Now I want to say this, just a reminder, when we are talking about osmosis and it involves plant cells, you are going to compare, the concentration of one solution to the concentration of the cell sap. So you cannot state something that goes like this. Solution B was hypertonic to the Tradescantia cells, to the cell sap of the Tradescantia cells. So the cells lost water to solution B by osmosis, became plasmolized, causing a decrease in length. Part C, apart from the length, name any other observation made on the stem in solution C. So these cells, the ones that were placed in solution C gained water, and that is the reason why we have an increase. Now by gaining water, the cells will become turgid. Therefore, the other observation is that the stem will be firm or rigid, because of the absorption of water. Question number 13. Study the diagram below and answer the questions which follow. Part A. Identify the muscle represented by letters A and B. So A, those are the biceps. B, those are the triceps. Now, just something, that I would like to make a note of. In most cases, when we see an S that is added at the end, it implies plural, right? But in this case, when we talk about the biceps and the triceps, that is actually the name itself, So you're not allowed to say bicep or tricep, no. They are biceps and triceps. So in plural and singular, the same thing. Part B, name the joint C, that is the hinge joint, the one found at the elbow. Part C, name parts labeled D, E, and F. So D, that is the olecranon process, the extension that is found in the ulna. E, that is the ulna, and F, that is the radius. Our next question. State the effects of oversecretion of thyroxine hormone. It increases metabolism, also increases the heartbeat, It causes enlargement of the thyroid gland, and Moving on. Describe the mechanism of closing the stomata on the basis of photosynthetic theory. So closing of the stomata occurs at night. At night, we don't have light. So no photosynthesis occurs. Glucose that is present is going to be converted into starch, which is osmotically inactive. Therefore, the osmotic pressure of the guard cell drops, causing them to lose water to the epidermal cells by osmosis. The guard cells become flaccid and draw close to one another, closing the stomata. Question number 17. An investigation was carried out on a mammalian kidney. So we have our kidney. We have an organ located above the kidney that is labeled T. That is going to be the adrenal gland. Now we have a pair of adrenal glands, each located above the kidney. So we have one on the right kidney and another on the left kidney. Now adrenal glands secrete hormones. For example, they secrete adrenaline hormone. They also secrete another hormone called aldosterone. Part B. What is the function of the structure named in Roman 1 above in the kidney? So we are talking about the role that the adrenal gland plays in the functioning of the kidney. So this is going to be tied to the aldosterone hormone. Now this is a hormone that is used for reabsorption of sodium ions in the kidney. Now, if a person, for example, has a low concentration of sodium ions, the adrenal gland would secrete more aldosterone that would increase reabsorption of sodium ions and causing an increase in the amount. If a person, for example, has excess or more than enough sodium ions in the body, less aldosterone will be secreted, therefore less reabsorption of sodium ions would occur in the kidney. So essentially it regulates reabsorption of sodium ions in kidney, maintaining the concentration, perfect concentration. Okay, part C, identify the disease shown in the kidney above. Now I know it might not be clear, but this is actually kidney stones. Part D, giving examples, state ways in which biological nitrogen fixation is carried out. So nitrogen fixation is carried out in two ways: biological nitrogen fixation that involves living organism, and the non-biological way that involves lightning. Now, biological nitrogen fixation can occur through symbiotic, bacteria. These are, for example, the Rhizobium species that are present in the root nodules of leguminous plants. It can also be undertaken by free living bacteria, for example, Azotobacter and Clostridium. Our next question, the diagram below represents a certain organism collected by a student at the seashore. Let's look at it. A pair of wings, 3 pairs of legs, ding dong, that is an insect. We can also clearly see that the body is divided into 3 parts. Name the class to which the organism belongs to, that is Insecta, starting with a capital I. B, state the observable features that enable the above organism to adapt to its habitat. Number 1, presence of the exoskeleton to support the insect above ground and also for protection against predators. Presence of legs, it has 3 pairs of legs to search for food, escape, unfavorable conditions. And it also has wings that enable it to fly and colonize new habitats. In fact, Insecta is the class that is the most successful in all, We have more insects than any other class in the world. Question number 19. What is the importance of seed dispersal? Number 1, it allows plants to inhabit new habitats. It prevents overcrowding. It also prevents competition for nutrients, space, light, etc. Our last question. The diagram below represents a section through the mammalian ear. Part A. Name the structures labeled R and T. R, that is the eustachian tube. T, those are the semicircular canals. Part B. State how the structure S, is adapted to its function. So that is the cochlea. So number 1, it contains perilymph and endolymph. These are fluids that conduct sound vibrations from the oval window to the sensory cells. It also has sensory cells that are stimulated by vibrations to produce nerve impulses. And of course, these nerve impulses are the ones that will be relayed to the brain. Part C. State the functions of structures Q and T. So Q, that is our pinna. It collects and concentrates sound waves into the external auditory meters. T, where are you? The semicircular canals, they maintain body balance and posture. And that brings us to the end of our lesson. By the way, I will be discussing paper 2, of Alliance High School quite soon. So be sure to check in and don't miss out. I will see you there.